Good afternoon. My name is Jeff Summers, and we are really happy that you are apparently in room 356 for the ultra-rare cancer development discussion session. We would like to thank AACR for the opportunity to speak today on an issue that's really important on the fundamental level to each of us that's here. And based on a number of the sessions that I've attended so far, this really seems to be a timely issue. And I'm sure that we are all sorry that we can't be there in person, and me particularly, as I've been invited to have a beverage with a number of you already. And unfortunately, I imagine in New Orleans and with the federal gift limitations of something like $10, that equals about a half a pint. So we have a number of pre-recorded talks, and that will be followed by a live and no doubt lively discussion, and there should be ample time for the audience to use Slido to ask questions. So without further ado, let's key up those presentations and move on. Good afternoon. My name is Jeff Summers, and I'm an Associate Director for Translational Sciences in the Office of Oncologic Diseases and CDER at FDA. I hope this afternoon's session will be enlightening. I know the speakers are each passionate about their roles in cancer therapy development for ultra-rare oncology indications. I work for FDA and have no conflicts of interest. The thoughts presented here today do not reflect official FDA regulations, guidance, or policy. The intent this afternoon is to highlight the challenges in developing for ultra-rare oncology indications and to discuss potential means to successfully confront those challenges. FDA, or at least the Orphan Drug Act, defines a rare disease as a disease or condition that has a prevalence of less than 200,000 people in the United States. This definition covers a sizable part of oncology indications. The Orphan Drug Act provides for various incentives, including tax credits, waivers of drug user fees, and market exclusivity. Many oncology indications that fall under the rare disease definition have thousands to tens of thousands of new cases per year. Besides the academic interest and clinical drive to improve patient outcomes, there's also an economic incentive to develop these drugs. This is not the focus of today's discussion. Although there is no accepted regulatory definition of ultra or nano-rare oncology indications, we are going to use a loose definition today derived from the sentiment of multiple drug development venture capitalists. There are less than 200 to 300 patients per year, we are not likely to fund it. Now that definition would be for non-curative treatment. As the larger the treatment effect, the greater the societal value of the drug, and that dynamic plays into acceptable pricing considerations, something that is far outside of my expertise and FDA's purview. And in this short time available here today, I am going to oversimplify decades of academic inquiry and attempts at improving the treatment for these diseases. The Oncology Center of Excellence at FDA has developed a program called Project Catalyst under the direction of Mark Thierry that is intended to provide support for small companies and academic accelerators developing anti-cancer therapies. Project Catalyst, in conjunction with the Pediatric Oncology Program and Rare Cancer Program in the OCE, under the directions of Greg Riemann and Martha Donahue, have endeavored in ongoing efforts to help facilitate drug development in these ultra-rare indications. There are multiple other government programs and patient advocate groups who are also focused on rare oncology indications, and some of these groups are here to present at today's session. We hope this session can provide awareness, impetus, and direction for undercapitalized, small patient population development programs. A quick roadmap for this afternoon. We will briefly describe the burgeoning malady defined as the following. Ultra-rare oncology indications have limited economic incentives driving development. Science is continually subdividing tumor types into smaller patient subsets that can become ultra-rare indications. Potentially effective therapies designed to target specific biology in ultra-rare indications may not be pursued. We will highlight some examples of the biology, tumors, indications, and ongoing subdivisions that reflect the need for creative and collective action. But first, some quick introductions. I have been summarily impressed from the interactions I've had with our speakers and am excited to hear their thoughts. Jeff Turetsky, a pediatric oncologist at Georgetown University, will discuss his experience in developing a drug targeting the Ewing sarcoma chimeric transcription factor. Josh Sommer, executive director of the Chordoma Foundation, will discuss efforts at targeting the transcription factor brachyury. Don Lowe, Division Director of Preclinical Innovation at NCATS at the NIH, will guide us through potentially expedited and resource-sparing drug development pathways and processes. Then Depanarian, an NCI SBIR program director who funds and mentors life science startups, will discuss programs inside and outside of NCI that can facilitate development for targeted treatments in ultra-rare tumor indications. And finally, Michael Stebbins, a former assistant director in the White House Office of Science and Technology, will discuss some high-level creative policy ideas that could address some of the issues with market failure drugs and indications. The problem or the challenge. There have been known pathognomonic oncogenic driver mutations in numerous tumors, with pediatric oncology being the exemplar poster child for 30 years. With unequivocal targets in small patient populations, the financial incentive for developing therapies targeting these pathway perturbations is limited. As the granularity of various omics technologies advance right down to single-cell transcriptomics, what were once tumor indications with large numbers of patients continually get divided into biologically different subgroups with potentially targetable pathways either now or soon. These potentially tractable targets, but now with limiting dilution subgroups, can become non-economically feasible indications to pursue, like our pediatric poster child. The question to ask, how can we facilitate oncology therapeutic development when the unmet medical need lands on an ultra to nano-rare indication? Even today, this moment in oncology therapy development, the hope and potential advantages of precision medicine can be lost when personalized medicine becomes commercially non-viable, individualized medicine. How do we as stakeholders, and unfortunately 40% of us who will also be patients, take a step forward together to facilitate, encourage, and incentivize development of these drugs and therapies? The next 6 slides are intended to highlight a key tenet of oncology that is imperative to surmount for any cancer. It demands, let alone defines, the necessity for developing therapies targeting these pathways or epitopes in ultra-rare oncology indications. Biology is complicated. There are a lot of perturbed and deranged processes that go on in a cancer cell. The problem is not finding targets. There is an abundance of potential targets involved in various aspects of neoplastic behavior. The problem is finding a target that has an overall impact on cancer cell survival in a particular tumor type, let alone generalizable across tumor histologies. This is a difficult task. Why? 1, different tumors have different biologies. 2, each tumor type can be subdivided into multiple different omic signatures, engendering different biology. And 3, cancer is evolutionarily gifted. Each individual patient's tumor has hundreds, if not thousands, of different biologies. A hallmark of malignancy is a mutational phenotype that results in tumor heterogeneity. This is the reason curing gross metastatic disease today is difficult and exceedingly difficult in today's oncology practice. It's a numbers game that was so eloquently articulated at the AACR annual meeting a number of years ago by Bert Vogelstein. We can cure tumors in mice, but not so easily in women and men. A 1.2 centimeter diameter tumor, approximately 1 cubic centimeter, contains 100 million to 1 billion cells, depending on whether they are epithelial or mesenchymal in nature. And the volume of a sphere increases exponentially by the radius cubed. Double the diameter of the tumor, and there are about 1 billion to 10 billion tumor cells. Now throw in multiple metastatic lesions and micrometastatic disease, and you can easily imagine a disease burden of 10 to 100 billion tumor cells. I apologize that this is Oncology 101, but we cure metastatic disease today with adjuvant therapy, both in adult and pediatric oncology. It's called micrometastatic disease, and even then we only cure some of it. How much is too much metastatic disease? If you can see it, it's probably too much disease. The hallmark mutational phenotype of malignancy, in conjunction with tens of billions of tumor cells, an inadequate cell kill from a limited number of effective drugs, results in selection pressure and clonal evolution. The paradigm in oncology drug development was frequently more is better, get to the maximally tolerated dose. This is slowly changing in the targeted era of today, and OC has a whole program called Project Optimus intended to help in this process. I mention this here because more is actually better. More agents and modalities that have non-overlapping mechanisms of action for efficacy is how you overcome the numbers game. It's how you win at the table, how you cure gross metastatic disease. We previously had surgery, radiation therapy, and classic cytotoxic chemotherapy that targeted rapidly dividing cells. Now, with the advent of agents targeting specific tumor vulnerabilities, other than just dividing fast, and the ability to manipulate the immune system to recognize tumors, the odds are improving against the house of cancer. Now, pediatric oncologists have a general philosophy of throw everything under the kitchen sink up front at gross metastatic disease, defined as many of the most effective drugs for that tumor given in close temporal association, as you get one chance to cure. I realize medical oncologists many times use a sequential approach for metastatic disease to extend life, because the kitchen sink approach did not work in the past and was toxic. As we develop more targeted therapies based on biology in conjunction with immunotherapy, I think the adult oncology sequential mindset may shift more towards the kitchen sink mentality. Oncology 101 lesson over. To illustrate the growing malady for today's discussion, we will briefly highlight 4 examples of pathways, some of which have been known for 30 years, that are unequivocally involved in tumor genesis and maintenance of the neoplastic phenotype. These are pathognomonic molecular events. These are targets searching for drugs. These are the poster children of ultra-rare oncology indications. And there's a plethora of additional genetic examples along the same lines. And if you think this conundrum is limited to esoteric cancers, in my opinion, the dawn has just begun, as we continue to spawn new poster children with ultra-rare indications from primarily 2 mechanisms. 1, subdivision of large tumor types into molecularly defined omic subtypes with potentially distinct vulnerabilities. And 2, the attainment of using HLA-restricted tumor-specific and tumor-associated antigens effectively in immunotherapeutic approaches. And the last postulate of our argument for the burgeoning malady, some setting every cancer patient into a pico-rare indication. All of us carry our own baggage through life, distinct places we have been and events experienced. They may not define who we are, but they certainly paint us. Each of our individual distinct cancers carries similar baggage, non-driving, unnecessary, colorful mutations that the advent of immune checkpoint therapy has shown to be highly useful patient-specific tumor vulnerabilities. Individualized, patient-specific neoantigen immunotherapy. The first 3 examples of targets with no drugs are all chromosomal translocation-induced chimeric transcription factors or transcription cofactors. These cancers occur in approximately 200 patients or less per year in the United States. Transcription factors have been notably difficult to drug because they have a relatively large interaction interface between the protein domains that recognize sequence-specific DNA. It has not been easily amendable to small molecule drug inhibition. However, these targets are becoming more tractable using creative approaches to drugging. Unfortunately, for the sake of time, we have to skip 30 years of biology here. The translocation in Ewing's was first described in 1984, and the EWS-FLY1 chimeric transcription factor gene was cloned in 1993. Alveolar rhabdomyosarcoma is characterized by chromosomal translocation involving the PAX3 or PAX7 transcription factors fused to the FOXO1 transcription factor, also cloned in 1993. An epithelioid hemangioendothelioma, a vascular tumor, contains a translocation-induced fusion of 2 different transcription cofactors, TAZ and CAMPTA1, first described in 2001 and cloned in 2011. An enormous oversimplification of the biology of the previous 3 translocation-induced chimeric transcription factors or cofactors is that altered chromatin structure and dysregulated gene expression ensue, resulting in oncogenesis and, importantly, persistence of the neoplastic phenotype. Important take-home point, 2 of these drivers have been known for 30 years. There are means to drug transcription factors today. PROTACs and molecular glues can target transcription factors for protein degradation. Protein-protein interaction inhibitors, or molecular disruptors as I like to think of them, can inhibit the homo or heterodimerization of transcription factors, thereby preventing DNA binding. Josh Sommer will discuss brachyury, which I know functions as a dimer and may be amendable to dimerization disruption. In addition, protein-protein interaction inhibitors can also inhibit co-transactivator interaction with the transcription factor, which Jeff Turetsky will discuss. And some of the protein-DNA binding domains have pseudo-like pockets that could be drugged, for example, the STAT proteins. So from transcription factors to our fourth poster child example, a more easily druggable chimeric kinase. Here we will discuss a tiny bit of the science as it brings home another point. Fibrolamellar hepatocellular carcinoma occurs in adolescents and young adults without pre-existing liver disease. A 400 kb deletion on chromosome 19 results in a chimeric fusion kinase that is constitutively activated because it no longer interacts with the pKa regulatory subunits. We recently had the pleasure to speak with Stephanie Shalm, who did the following work at Blueprint Medicines. What's great about academicians and even small companies is that they are much more likely to pursue interesting science and targets that have limited prospects for commercialization. In a paper that was recently published, they showed using a patient-derived xenograft fibrolamellar carcinoma model that the chimeric kinase was necessary for maintenance of the neoplastic phenotype using shRNA downregulation. Blueprint Medicines went on to make two compounds with exceptional drug-like properties, with IC50s of 1 nanomolar and 0.3 nanomolar, and the IC50s for closely related kinases were 80 and 40-fold higher. And in initial activity studies using suboptimal dosing, Q24 hours, that allowed PK activity to rebound to normal levels, there were still notable decreases in PDX tumor growth. These were potent and specific small molecule drugs that hit an unequivocal target in the genesis and maintenance of fibrolamellar carcinoma. BPM decided not to pursue further development of these compounds as they also inhibited wild-type pKa and were thought to have too much potential for cardiac toxicity, data which I have not seen. They did not enter first-in-human studies, and this was probably a reasonable commercial decision. I think this happens. Hypothetically, do I see a straightforward approval pathway, let alone a commercial opportunity, for a drug in an ultra-rare oncology indication that is toxic enough to be limited to a few courses of therapy that simply generates a response with no substantive duration of response in a single-arm trial? No. However, I do see a targeted drug for this ultra-rare tumor, albeit at the cost of increased toxicity. An additional 2-log kill as a component of multi-agent therapy might be the crux move to curative intent someday. Unfortunately, there is little to offer in this malignancy to produce the first 9-log kill. Now you may say those are just isolated examples, Dr. Summers, but there are a lot of these examples, particularly in pediatric oncology and sarcomas. But to bring the point home to general adult oncology, as the techniques to characterize the omic signatures of neoplasia advance, there is an ongoing subdivision of major tumor types into different biologies. I recently heard a talk by Stefan Pfister where his team identified 200 molecularly distinct brain tumor types by methylation epigenomics. And as he points out, and is the whole point of this afternoon's discussion, the treatment approaches by far do not match this molecular complexity. This was a meta-analysis of a number of papers performed by Serafini and Restaghini, in which they defined 60 different omic subtypes of head and neck cancer. Now, to be fair, there have been successes. This is a listing of some of the genetic alterations in non-small cell lung cancer, many of which have been targeted successfully, partly driven by the initial large patient population, such that the subgroups don't result in ultra-rare indications, and the generalizability of some of their targets to other potential indications. And you can look at the omics challenge as a glass half full, glass half empty kind of thing. The improvement in various omics technologies continue to molecularly define major histologic tumor types into smaller subsets. Glass half empty, potentially too few patients. These subsets are more biologically homogeneous with subgroup-specific dependencies and cellular vulnerabilities underlying tumor genesis. Therapies designed to target homogeneous biology will be more likely to produce substantive activity. Glass half full. Now let's transition to my favorite topic, immunology, and the second mechanism whereby ultra-rare oncology indications are generated. The attainment of effective HLA-restricted, tumor-associated, and tumor-specific antigen-based therapies. There are not many highly tumor-associated, let alone tumor-specific antigens that are expressed on the plasma membrane that can be targeted by conventional antibody-based approaches. The ones that are approved have their targets listed here. The advantage of these plasma membrane-expressed tumor-associated antigens is they all look the same in each of us. Most of the mutated tumor suppressor genes and oncogenes, as well as oncofetal differentiation genes, are intracellular proteins that are sequestered by the plasma membrane from antibodies. But the beauty, the pulchritude of the immune system, the elegance of antigen processing and presentation by the MHC system allows many of these tumor-associated and tumor-specific antigens to be recognized as peptides presented by HLA molecules. Unfortunately, they can look three-dimensionally different from person to person depending on their class 1 repertoire, subsetting the patient population that any specific peptide MHC therapy could be effective for. There are a number of HLA-restricted therapeutic approaches listed here, but one of the most exciting approvals in the past couple years in oncology, at least from a mechanistic perspective, and that highlights the potential and limitations of these approaches, is tebentafusp. Tebentafusp is a bispecific engager utilizing an engineered soluble T-cell receptor targeting an 8 amino acid peptide of the tumor-associated differentiation protein GP100 presented by HLA-0201, while the other end recognizes CD3 on T cells using a single-chain antibody variable fragment. When I first heard this presented as a new IND review several years ago, I seriously said no way and honestly questioned if it could actually work. Well, it did. It prolonged overall survival in metastatic uveal melanoma and was recently approved. As you can see by the number crunching, there are approximately 2,500 cases of uveal melanoma in the U.S. per year. Not all of those are initially metastatic, but a guesstimate that over 10 years, at least 30 to 40% become metastatic. And then include HLA-0201 restriction element, and the eligible patient population is probably 500 patients per year in the U.S. Choosing any other of the most highly expressed HLA molecules in the U.S. would decrease that to less than 200 patients. Now, to bring the point home on how quickly HLA-restricted approaches can subset an indication, this was a fantastic paper published last year in Science by Sui et al. In this case, instead of using a soluble T-cell receptor as in tebentafusp, they employed an antibody-based T-cell receptor mimetic that recognizes a specific point mutation in the p53 tumor suppressor protein presented as a peptide on the HLA-0201, and the other end recognizes CD3 on T cells. A quick back-of-the-napkin calculation guesstimate. You figure there are about 1.8 million cancer diagnoses and 600,000 deaths in the U.S. per year. p53 is the most commonly mutated tumor suppressor gene in cancer at around 40%, but there are probably 100 different mutations, although a third of them are reflected in about 8 different mutations. The R175H mutation is about 5.6%, and this peptide is restricted by the most favorable HLA-0201 allele in regards to frequency. But in the end, that comes down to maybe 4,000 patients, which is plenty. But imagine the development plan to demonstrate efficacy across heterogeneous tumors and varied natural histories, and possibly the necessity of an overall survival endpoint based on mechanism. Now pick a different p53 mutation presented by a less frequent HLA molecule in the US population, and the number of patients decreases substantively. And the development plan requirements, probably not so. And finally, the ultimate mechanism in generating pico-rare, individualized oncology indications. Cellular-based neoantigen approaches. These are approaches in which patient-specific neoantigens are identified by next-gen sequencing and T-cell clones generated towards those neoantigens. It's patient-specific, resource-intensive, but has shown activity in metastatic epithelial cancers. It's literally taking the mechanism of the paradigm-changing PD-1 approach and operationalizing it at the cellular level, and is potentially applicable for even tumors with low mutational burdens. The intent in these short minutes was to communicate that our ever-advancing ability to interrogate biology, if not already, will soon define cancer as 1,000 diseases. And those 1,000 diseases will require hundreds of specific treatments to cure metastatic cancer. And many of these will not be economically attractive from a commercial standpoint. I'm excited to hear the creative ideas that our speakers and panelist members have on addressing what many see as a formidable challenge. I'd like to thank Mark Thierry, Martha Donahue, Julie Schneider, and the OCE for advice and guidance on this presentation, which is greatly appreciated. And this is a slide with some links on some of the programs that were mentioned in the talk. And I thank you very much.
I would like to thank the organizers for allowing me to share my story today about how we try to commercialize a drug for a very rare cancer Ewing sarcoma. First, my disclosures that small molecules that I will show in this talk are patented, and this intellectual property has been licensed by Nocturnal Therapeutics. I'm a founder of this company and hold equity shares. Also, I will not be discussing any off-label uses, as this is still an experimental agent. Ewing sarcoma is a very rare malignancy affecting around 500 patients a year in North America. And as you can see from this survival plot, even though it's almost 20 years old, survival for metastatic patients has not improved significantly in that time. Patients with localized disease saw an improvement with the addition of ifosfamide and etoposide, and with interval compression, that survival has improved a small amount, but we still have a long way to go, and patients who do survive have a lot of toxic complications. So we'd like to be able to figure out a drug that would be both improving survival with less toxicity. In order to do that, we would want to have an appropriate target, because if you have a good target, you can improve the therapeutic index and therefore improve survival without impacting toxicity. Ewing sarcoma was named after James Ewing, who described the tumor just over 100 years ago. And in 1992, the fusion protein EWS-Fly was identified at the 1122 breakpoint, which had been identified about 10 years earlier. And it turns out that using various antisense technologies, we knew that if we could get rid of EWS-Fly, you could inhibit the growth of Ewing sarcoma cells. But using antisense was a big challenge, because getting systemic treatment is still beyond our ability. So it took a deeper look at EWS-Fly in order to start to think about how to solve this challenge. And what you have here is the EWS portion of the fusion, this first half of the protein. And this part, this red part, initially was thought to be simply a transcription activation domain. We now know that the fusion protein functions wholly different than either of the parts. And the Fly1 part includes this DNA binding domain, this ETS family domain. And one initial way one could think of targeting this is to block the fusion protein from binding to DNA, but unfortunately the specificity of that is poor. And if you start non-specifically blocking things, you end up with drugs like doxorubicin with a lot of non-specific toxicity. So here we have this fusion protein. It lacked a kinase domain at the time, which was thought to be one way to target proteins and inhibit them. And so we had to be very creative with how we decided to target this. So we knew that it would require a little something imaginative. So just as a point, imagine a color you've never seen before, and that's pretty much where we started 20 years ago to try to target EWS-Fly. We had this idea that if you could figure out what the proteins were that interacted with EWS-Fly that were critical for its function, we knew it bound to DNA. One protein we identified was RNA helicase A, and the idea would be that if you could take and create something that would bind to EWS-Fly and disrupt an interaction, like a small molecule that would stick to EWS-Fly, block the binding of RHA, and potentially then kill the tumor. So we use surface plasmon resonance as a way of identifying such a small molecule. And the initial molecule we identified was from this NCI set. And then we derivatized that. Yali Kong, the chemist, created this version that has a methoxy here instead of the chloride. Otherwise, they're identical. And we knew that this molecule could bind to EWS-Fly and block the interaction of RHA. We just didn't know where on EWS-Fly it was binding. However, one of the features of this molecule, which I'm sure you've noticed by now, is a chiral carbon. That chiral carbon allowed us to separate EWS-Fly into R and S enantiomers. And that allowed us to go on to show that the S enantiomer was significantly more potent at inhibiting growth than the R enantiomer. The S enantiomer blocked the interaction of RHA, whereas the R enantiomer did not. The S enantiomer, like the racemic, inhibited transcription, whereas the R enantiomer did not. And more importantly, in this direct protein assay, we show that RNA helicase A is very potent, rapid unwinder of double-stranded RNA from double to single-stranded. If you add in EWS-Fly, that unwinding is inhibited. Now, if you block the binding of EWS Fly by adding SYK4269, you see the inhibition is removed and RHA rapidly unwinds the RNA. And if you add in the R enantiomer of YK4279, you do not block the interaction and the unwinding is inhibited. We also showed that in a rat model of Ewing sarcoma, that you could start with tumors that were around 2.5 cubic centimeters, and if you treated the animals for enough time with continuous infusion, you could get complete regression, as shown here by this biopsy of the leg where the tumor was, where you don't see any of the CD99 positive cells. We showed that in this study there were 2 animals that had complete regression, 3 animals with stable disease, and 1 animal grew, and these are the control animals in black. So that led us to essentially try to commercialize the drug. And in 2009, we originally reported the YK4279, and that almost immediately led to families and patients getting in touch with us wanting to use YK4279 to treat their loved ones. We tried to license YK4279 to a number of large pharmas and medium-sized pharmas, and none of them were ultimately interested. They did a significant amount of moving the goalposts when we would share data with them, but at the end they decided that Ewing sarcoma wasn't a significant enough disease to target. So that left me with a challenge: either give up on this or create my own company. So I tried twice to create a company. The first one was called Targeting Disordered Proteins 1, and that failed because it had company leadership that didn't know what they were doing. Then we created Targeting Disordered Proteins 2, and that also failed, also because the company leadership didn't know what they were doing. And it turns out that you really need somebody that knows how to start a company. And in 2013, Scott Glenn reached out to me about a different project. We talked about the YK4279 for a while, and in January of 2014, Scott provided seed funding by contacting previous investors in other companies he had started, and Tokelis, Turetsky, Karen, Lauren, and Scott was born. And eventually that led to an analog called TK216 that the company created. And in January 2016, the IND was granted by the FDA. We showed that TK216 had the same enantiospecificity as YK4279 in many assays. Here we show it blocking RHA. We then went on to show toxicology in 2 species to show that the drug could be safely given to humans, or at least started to be. And we started our phase 1 dosing very, very low, down at 18 milligrams per kilogram, eventually going up to a dose-limiting toxicity of neutropenia, and eventually going to an expansion cohort. Now, we knew we had to give the drug for a longer period of time because of pharmacology, and when we reached that magic 14-day infusion time, we did notice that a patient who had multiple regimens for Ewing sarcoma, who was 19 years old at diagnosis, presented with 4 pulmonary metastases. And after 2 cycles of TK216 alone, 3 of the 4 were completely gone. The 4th one lingered. 6 months later, he had surgery to remove the 4th nodule and continued on TK216. After 2.5 years from starting TK216 and 2 years from the time he had surgery, they decided to stop the drug. And now, 6 months later, almost 3 years after starting on the drug, he remains disease-free. This patient had multiple pulmonary nodules a year and a half after completing standard Ewing sarcoma therapy. He went on TK216 along with vincristine as a synergistic agent, and all of the nodules eventually disappeared by cycle 6, and he remains on treatment over 2 years later, completely without any evidence of ongoing disease. And the phase 2 trial is continuing at the present at that dose level. So Oncternal Therapeutics was created because the company brought in an antibody against ROR1 and another agent later, and also with clinical trials. And one of the challenges that we've discovered is that, of course, not all patients are responding. So one of the questions we're trying to get answered now is why some patients have responded to this, in theory, very targeted drug and others haven't. We also learned that many other cancers might be targetable with TK216, including lymphomas, work that we did in collaboration with Francesco Bertoni, and prostate cancer, work that we did in collaboration with Georgetown colleague Aykut Eren. Work that was done by others, not collaborative with us, also affirmed the drug was effective in Ewing sarcoma. Two groups showed that it was effective in neuroblastoma, and more recently a group targeting ETS proteins showed that it was effective in inhibiting melanoma. So while we never set out to either create a company, get rich, or have any significant financial gain from this, I am nevertheless pleased and with the results for a couple of patients that basically is summed up here by this paraphrased quote from the Talmud: "If you save one life, it is as if you saved the whole world." Now, we know that patients with Ewing sarcoma continue to need more effective, less toxic therapy. We know that patients with other cancers may benefit, and we need to solve the challenges of EWS-Fly interaction and patient resistance to therapy. I want to first start by thanking all the patients and their families who trusted TK216 and Oncternal in their care. Other key collaborators included Julie Foreman-Kay in Toronto, Steve Metallo in GU Chemistry, Milton Yali did the original chemistry at Oncternal Therapeutics, James Breitmeyer, Gunnar Kaufman, and Salim. And really, most importantly, Aykut Eren, who's been a colleague of mine for over 20 years and has been along for this ride and been critical in the work that we've done, along with a number of trainees, both at the postdoctoral and graduate and undergraduate level. The work also required a lot of philanthropic support to get going. The Children's Cancer Foundation was critical for that. Other foundations, as shown here, have come along and made important contributions to the work. And once things really got going, we were able to get significant funding from the NCI. And of course, now Oncternal is carrying this project forward, along with additional work, including an SBIR grant from the NIH. I'd like to stop at this point. Thank you for your attention. Again, thank the organizers, and again, thank all the patients who were involved in this trial, and look forward to participating in a discussion.
Hi, my name is Josh Sommer. I'm co-founder and executive director of the Chordoma Foundation. I'm going to be talking about our efforts to catalyze the discovery of therapies against brachyury, which is a very important target for chordoma, as well as perhaps several other tumor types. I have no financial relationships to disclose. For me, this is a really personal mission. It started about 16 years ago when I was diagnosed with a skull-based chordoma and became determined to try to change the odds and bring about better treatment options for those of us affected by this disease. Initially, that led me to join a lab at Duke, where I was a student at the time, to try to advance research that way, and ultimately to partner with a number of other patients and family members to start the Chordoma Foundation to work towards a better future for all of us affected by this disease. Ultimately, that snowballed into a global movement involving thousands of patients and family members worldwide, as well as doctors, researchers, companies, and funders as well. Together, we've made a lot of progress. We've catalyzed research across the entire R&D continuum, from developing models of chordoma to understanding the biology of this disease, and ultimately letting that lead us towards clinical trials. Thankfully, there have been a number of tractable therapeutic targets that have been identified, and that's led to a number of drug repurposing opportunities. But perhaps most importantly, the research has led to the understanding that chordoma is really driven by this transcription factor, brachyury, which seems to play an initiating role in chordoma pathogenesis, but is also a key essentiality, and in fact, not only essential, but the most selectively essential gene for chordoma. And indeed, when you knock down brachyury or eliminate it through degradation in chordoma cells, that leads to profound growth arrest and a senescence-like phenotype. So a lot of evidence to suggest this could be a very, very important target for chordoma. On top of that, it is only transiently expressed during development and is largely absent postnatally, which we believe provides reason to believe that there could be a wide therapeutic window for brachyury drugs. So really, really promising opportunity, however, definitely not without challenges. For one, brachyury is a transcription factor and so widely assumed to be undruggable. It is definitely the case that chordoma is an understudied disease, and so perhaps that raises questions about the feasibility of actually bringing drugs into the clinic for chordoma patients. It is definitely an ultra-rare cancer, only about 300 new cases diagnosed per year in the United States, a total market probably of about 2,000 patients in the US and Europe at any given time with advanced disease who might be in need of systemic therapy. So certainly not a large market to incentivize investment in drug development. But we believe fervently, and we're starting to see evidence that these are all surmountable challenges. And I'm going to talk now about some of the progress that's been made in overcoming each of them. First, it's been incredibly heartening to see how over the last decade or so there has been a convergence of advances both in new therapeutic modalities as well as enabling technologies, which really for the first time make it plausible and perhaps provide a line of sight to therapies against transcription factors. And we've been really, really grateful to be able to partner with the Mark Foundation for Cancer Research to support several different proof of concept projects aiming to bring those approaches to bear on brachyury, perhaps as an exemplar of what's possible for transcription factors more broadly. And much to our delight, it's turned out that, in fact, there are quite a number of potentially druggable pockets on this protein. And that has now served as a jumping off point for a number of hit to lead projects, both pursuing functional inhibitors as well as monovalent and bivalent degraders. At the same time, we've worked to develop a number of enabling resources that are going to be critical for preclinical development of any brachyury therapy, regardless of modality, everything from purified proteins to reporter assays, and certainly the preclinical models that will be needed to test those drugs for chordoma. It's one thing to make those reagents available, but it can still be costly and time consuming to actually run those assays. And so to grease the wheels, so to speak, we've developed a number of centralized research capabilities, starting with in vivo drug testing as well as in vitro drug testing. And now in total have tested almost 70 concepts in chordoma PDX models. Most recently, we've actually started a lab of our own to expand the breadth of capabilities and be able to run a wider variety of cell-based assays, both to pursue internally generated hypotheses, but also to support the discovery biology, target validation, and translational research efforts of various academic and industry partners. And the idea here is that by centralizing these disease-relevant assays, we're able to benefit from economies of scale to drive down the cost of running these assays, to decrease startup time and increase the robustness with which they can be run. At the same time, we've worked to pave the way towards clinical development of therapies for chordoma, supporting the initiation of a number of clinical trials, perhaps most importantly by providing connectivity to the chordoma patient community. And in total now, we've supported and helped to initiate 7 clinical trials for chordoma, really demonstrating feasibility of clinical development for this disease. And while chordoma is the most obvious use case for brachyury drugs, there's a growing body of evidence showing that it's expressed in a number of other tumor types. It seems to be associated with poor outcomes, metastasis, recurrence, et cetera, and seems to underlie resistance to a variety of standard therapies, which all hints at the potential of a market for brachyury drugs even beyond chordoma. However, to validate that, it's really critical that we be able to probe brachyury's function in a variety of preclinical models representing other diseases. And to do so, the linchpin, the key missing piece is having high quality chemical probes. That also is really critical as a jumping off point or as a template to facilitate and de-risk subsequent lead optimization efforts and ultimately the development of clinical candidates. So a big focus over the last several years of ours has been to try to catalyze probe development. We've been very grateful and fortunate to partner with the Mark Foundation as well as the Marcus Foundation to invest in a consortium involving investigators at UNC, ICR, Oxford, and elsewhere. And this group has made incredible progress to optimize some of those weak initial hits from early screens that I mentioned previously, and has driven towards a number of chemical series with biochemical potency in the sub 10 micromolar range, and is driving towards probes of a variety of types, PROTACs, covalent, as well as reversible ligands that have in vivo activity in the sub-micromolar range. So we really believe fervently in the model that has been pioneered by the Structural Genomics Consortium and others in providing these tools to the research community with no strings attached to make them as easily accessible to the widest variety of researchers possible, to be able to use them to validate this target in other diseases, as well as to make them available to companies to serve as a starting point for lead optimization and ultimately development of clinical candidates. So I think what we've seen now is over the last several years, what has turned, what it was initially this quixotic and very seemingly difficult undertaking has given way to just a variety of opportunities, both for investigators to learn about brachyury's function, for other foundations perhaps to catalyze the validation of brachyury as a target for diseases of interest, and certainly for companies perhaps to demonstrate proof of concept for emerging technologies that could be applicable to transcription factors more generally, using chordoma perhaps as a test case and a de-risk path to market. So this work was only possible thanks to the investment and partnership and work of quite a number of partners, including the Mark Foundation and the Marcus Foundation. Collectively, we've invested over $3 million in the work described above, and undoubtedly there's going to be more philanthropic and public investment needed to get to the point where this target and the compounds that are emerging are sufficiently de-risked to become a viable business proposition. But I think we're well on our way to getting there. And certainly the just incredible efforts and fearlessness of the investigators who have taken on this daunting challenge and supposedly undruggable target. I think we're well on our way to overcoming the notion of undruggability. So I really appreciate the opportunity to share this story, and very much look forward to continuing a conversation with anyone who might be interested in joining in these efforts. Thank you so much.
Hi, I'm Don Lowe. I'm the next speaker in this session. I am an employee of the National Institutes of Health, and I have no financial relationships to disclose. What I would like to do is tell you about our efforts in this area that we're undertaking at the National Center for Advancing Translational Sciences, or NCATS. Most of you may know, but NCATS is the newest kid on the block. So we've only been around for around 10 years. One of the 27 institutes and centers of the NIH. And unlike the other institutes, we were set up without an explicit basic research component. And the reason it can really be summed up in this problem statement. That is, we've been done an amazing job over the last decades as a biomedical research community in understanding the molecular basis of diseases. But to date, there are only, say, around 500 therapeutics for these diseases that are FDA approved. And each year, there's really only a handful of new treatments for for novel diseases that are approved by FDA. Scannell, about 10 years ago, coined Eram's law, which essentially compounds this problem. That is, unlike Moore's law, which notes that the costs of new microchip production decreases exponentially with time. Eram's law notes that the cost of new drug development actually increases exponentially with time. So shown in Eram's law graph here is the number of new drug approvals per $1 billion of R&D spending. And you can see in the semi-law graph that this declines exponentially. Now, Eram's law and the cost of drug development R&D is really driven in large part by the spectacularly bad failure rate of new drug development. This is a graphic developed by Sun and Hu at University of Michigan, but you can find many such examples and analyses showing the dramatic and essentially 90% or more failure rate of new drug development, starting at the initial stages of target validation, identification, and the development of new drug candidates, and all the way through approval or lack thereof. So really, this is the context in which NCATS was set up. And its mission is really to tackle the common bottlenecks that continue to drive failures across the spectrum of new drug discovery and development, from the very beginning all the way up through FDA consideration for approval. The group that I'd like to tell you more about is called the Therapeutic Development Branch, or TDB. It's part of the intramural research program at NCATS, and we live in this red circle part of the chevron diagram, the common chevron diagram for drug development. So our work really starts around the time that a new lead candidate is identified. We can take that through lead optimization and through the entire suite of IND-enabling studies as required by FDA. Of course, IND clearance is required to initiate clinical studies, and this is quite a large-scale undertaking, usually several years and usually several $1 million. It turns out that there are very few large-scale funding mechanisms at a large enough scale to really fund IND-enabling studies, and because of the high risk, commercial investment in this space is also quite scarce. As a result, it's this stage of preclinical drug development is very often called the Valley of Death. So the Valley of Death is so called because if you look at this graph of the value of a new drug candidate, you can see that as you commit more resources and time in this pre-lead to lead candidate development phase, and you take this drug candidate through IND-enabling studies, the value of the asset actually declines. And there's really no positive value inflection until the drug candidate actually is taken through IND clearance into phase 1 clinical trials and really doesn't start to substantially appreciate until proof of concept of efficacy in phase 2A. This valley of death for rare and ultra-rare diseases, including cancers, is then further deepened and widened by the fact that the prevalence of rare and ultra-rare diseases is generally considered too low to be commercially viable by the commercial markets. And you can see in this histogram of the prevalence of diseases that really the vast majority of rare diseases are too low in prevalence to represent a viable market. So the NCATS notion was that, what if we could address this by providing the general expertise, which is missing in this area, partly as a result of funding scarcity. What if we could provide the resources and the expertise to wrap around new target and lead candidates from outside partners, let's say from academic labs or from small biotech spinouts? So instead of trying to require an academic or a small biotech company to go out and find the IND enabling resources and expertise you would need to put together an IND package. What if NCATS could partner with academics and small biotechs in a collaborative way to complete IND enabling studies and through to phase 1 and phase 2 clinical trials? So the way we've done this is to establish a standing team of around 60 scientists. Almost all of us are veterans of biotech and pharma across all the discipline areas that you would need to put together an IND package. So you can see in the org chart, we have biology, hydrobiology, and medicinal chemistry teams to really take a lead and optimize it into a viable drug candidate. The DMPK teams, safety, evaluation, toxicology, CMC manufacturing, formulation development, drug product manufacturing, and then a key component, project management, to keep all of these teams together on track. And to put these together with an outside academic or small biotech to answer all the questions that the FDA requires for a new IND filing, namely, do you have a drug candidate that is potent and selective enough? Can you demonstrate efficacy in a disease model? Can you show GLP toxicology and safety pharmacology? Can you develop a manufacturing method that develops enough of the drug substance in pure enough form to be then formulated into a GMP manufactured drug product that's appropriate for the route of administration proposed? Again, as I mentioned earlier, this is a multi-year process that takes millions of dollars to put together such an IND package. So using this partnering approach, we've managed to help clear 44 INDs today over the last decade. And most of these all are percolating through phase 1, phase 2, phase 3 clinical trials. We're hoping to get our first approvals this year and next year. You'll see maybe in tiny print on the left pie chart that in fact we, in the yellow pie slice, we don't work that much in oncology, partly because our bigger sibling, the NCI, is quite active in this space. But I'll give you a couple of brief examples of oncology projects we've taken on in this IND space, where the contribution that we made really was very specifically not in the basic science or in the development of the concept, but rather in putting together the IND packages required by FDA. I know we're running short of time, so I won't be able to tell you much. This first project is a collaboration with an academic, Dr. Jessie Au from University of Oklahoma and her spin-out company Optimum. And here the idea was that tumor penetration of chemotherapeutic drugs has always been a problem. So in this case, not inventing a new drug, but inventing a new formulation, a tumor-penetrating microparticle that could carry a very known and well-characterized payload, in this case paclitaxel, to carry this into tumors. And again, the challenge here was putting together an IND package for the tumor-penetrating microparticle. So this took several years, several $1 million, but the IND has been cleared and first-in-human studies have begun. A second project just to mention, this was a broader project involving a broad collaboration. You'll see in the upper right with 3 universities, University of North Carolina, Chapel Hill, Northwestern, and Kansas. Then together with our companion branch at NCATS, the early translation branch, to put together a phenotypic high throughput discovery effort to really target perinuclear compartment formations that are associated with metastatic cancers. And so although this is a very new area, the combination of academic teams, our in-house drug discovery group, the early translation branch, managed to develop a drug candidate called metarestin that was then shown to be effective in multiple metastatic cancer xenograft models, as shown on the left. Then our branch, as I had mentioned, has used this broad partnership model to take metarestin through IND-enabling studies. So DMPK, the GLP tox, safety pharmacology, very importantly, the scale-up and manufacturing formulation of the drug product through to regulatory filings with the FDA. So this has been cleared, and through COVID, actually, the phase 1 trial has been ongoing, first in human studies in advanced solid tumors. And this has been undertaken by Udo Rudloff, as shown on the upper left at the NCI. But we're very excited about this model in that the productivity has been compared to average in industry and academia, pretty high. And the hypothesis we're testing essentially is whether such a non-dilutive partnership model can really help to get exciting new drug candidates, especially for rare and ultra-rare disorders, through this preclinical valley of death, and thereby hopefully put better drug candidates into clinical testing with greater promise, and hopefully starting to make a dent in this big gap between the number of diseases we know and the number of FDA-approved treatments. So thanks very much for your attention. I'll be happy to answer questions in the question period at the end.
Good afternoon. I'm Deepa Narayanan, a Program Director at the SBIR Development Center at the National Cancer Institute. Today, I'm going to talk about the SBIR resources available at NCI to support small businesses in the cancer space. And in particular, I'll talk about 2 new funding opportunities that are active right now. One for early stage technology development for rare and pediatric cancer, and another one specifically to translate innovative technologies from university research labs to the industry. No financial disclosures. The SBIR and STTR programs are America's seed fund. It is the largest source of early stage capital available for life science technologies, with more than $1 billion available across the NIH. At the National Cancer Institute, we have about $182 million each year for supporting small businesses in cancer. The STTR programs are a smaller pool of money, and it is intended to facilitate technology transfer from universities to for-profit small businesses. In both cases, the award is always made to the small business. At the NCI, we have a centralized model of SBIR management, where we have a portfolio of about 400 to 450 active projects at any given time. About half of these are cancer therapy, and the other half is divided between imaging, in vitro diagnostics, research tools, and health IT. We have seen some progress in the recent years in the number of rare cancer technologies that we fund within the SBIR program, thanks to the FDA's Orphan Designation Program. However, we tend to get a lot of application in few types of cancers, and it's almost negligible in other types of rare cancers. This is disappointing, but it's not surprising given the complicated and time-consuming process it takes to take these technologies all the way to patients. Also, private investments in these cancers are low due to small patient population. So if the SBIR program can de-risk technology such that these companies can attract further private capital, then that would be considered a huge win for the SBIR program. To be eligible for an SBIR or an STTR award, the company should be organized for-profit United States small business concern at the time of award. The company must be majority owned and operated by US individuals and have less than 500 employees. In the case of SBIR, majority venture-backed companies can apply as long as no single entity owns more than 50% of the company. However, in the case of an SBIR, the PI's primary employment should be with the small business concern, both at the time of award and for the entire duration of the project period. The STTR has similar eligibility criteria, except that the PI does not have to be majority employed with a small business, but the company does require a collaborative agreement with a non-profit research institution such as a university. these programs set up? Now, the SBIR and STTR programs are considered to have three phases. The phase 1 component, and this is not clinical trial phase 1, this is SBIR. Phase 1 is generally a feasibility study, like proof of concept. The guideline for budget is about 300 to 400,000, and the project period is approximately about 12 months. The phase 2 is the full research and development phase, with the funding of a total of about $2 million, typically for 2 years. The phase 2B is a special NCI award for clinical validation and translation. This is the biggest award, amounting to about $4 million in 3 years. This funding should help the companies reach phase 3, which is the commercialization phase, where the companies can raise private capital funding to commercialize their technology. A company can apply for a fast track award where they submit both phase 1 and phase 2 together, or they could apply directly to a phase 2 program for an SBIR grant, and this can limit the time between grants. But these programs are competitive, and it is best attempted only if you have substantial preliminary data. At the NCI, we fund via both contracts and grants. Grants can be investigator-initiated, which means the applicant can apply for any technology as long as it meets the mission of NIH. Or grants could be targeted solicitation focusing on a particular area. Contracts are typically key NCI priority areas, and they have their scope of work defined by the NCI. Although we do like to keep it broad to encourage more innovative ideas from the applicants. Our contracts budget is usually between 10 to 25% of the total SBIR budget. While the intent of both mechanisms are fairly similar, there are differences in how they're structured and operated. I'm sharing a list of currently available funding opportunities. Most of our applications come in through the omnibus mechanism, which has 3 deadlines each year, on 5th of January, April 5th, and September 5th. However, we have other funding opportunities, but as I mentioned before, I'll focus on these 2 new funding opportunities that were released just 2 weeks ago. We have a brand new funding opportunity for pediatric and rare cancers that I'm very excited about. This is a unique funding opportunity that uses the SBIR contracts mechanism, and we are calling this our NCI Concept Award. The goal for this particular funding opportunity is to fund really early stage disruptive technologies. The applications that we get at NCI often require preliminary data to be competitive, and therefore the truly radical concepts do not enter into the program. This is our effort to remedy that and welcome innovative ideas without preliminary data into the program. So what's the Concept Award? The NCI SBIR will provide $300,000 for small businesses to fund experiments that can obtain initial de-risking or proof of concept data. In this pilot program, we are focusing on innovative and potentially transformative therapies, diagnostic tools, or preventative strategies focused on pediatric or rare cancers. Proposals do not require preliminary data at all, which is not the norm for SBIR, so this is truly unique in that concept. Some other unique features, in addition to not requiring preliminary data, are that there is a high consideration on innovation. 50% of the peer review score is weighted for innovation. The other unique feature is the ability to get programmatic input prior to submission by submitting a white paper. After review of the white paper, NCI will inform you if your solicitation meets the scope of the program and advise you either to submit the proposal or suggest other alternative funding strategies. The application is incredibly short. It's less than 20 pages. In fact, it's a fillable PDF proposal form, so that's pretty unique. Awardees are also given additional funds to go through an entrepreneurial training program called the I-Core, which refines their value proposition and business model. The application due date is August 22nd, 2022. So I mentioned that this was for rare cancers and pediatric cancers. The definition of rare cancers is a little complicated. So we're using the definition provided by the NIH's Genetics and Rare Disease Information Center. However, we do expect applicants to identify the exact population that they are trying to help and address how this proposed technology will make a difference to that selected patient population. I mentioned before that applicants can submit a white paper. The deadline to submit the white paper is May 2nd. Each white paper is assessed by 3 internal NCI experts to see if the proposed idea is within the scope of the original solicitation. And then each applicant or offerer is provided written feedback providing guidance for next steps. And another really critical feature of this application is the time to award. The last round, the awards were made in 5.5 months from submission. And for those that are familiar with NIH's application timeline, you'll realize that's about half the time it takes for our normal awards. The second funding opportunity that I'm excited about is not focused on rare cancers, but it's a very unique mechanism modeled on the K99R00 grant mechanism. This is our small business transition grant. One of the things that we realized after talking to several universities is that in many cases, it's the postdocs that drive the transition from academia to entrepreneurship when it is time to spin out a university startup. However, we also realize there are lack of resources to provide a mentored transition to industry and provide guidance to that postdoc. To help this, the program that we created is a fast track award. The phase 1 is in fact an STTR. So the postdoc, who would be the PI of the grant, can continue to be majority employed by the university. They will perform phase 1 work, where the proof of concept work is done under the guidance of perhaps the academic lab head. Once the phase 1 is completed, the program will evaluate if they have met the go, no-go criteria for transition to phase 2. The program will also ensure that the mentoring plan and the commercialization plan are still relevant. And once they transition to phase 2, the PI, who's a postdoc, will move to be full-time employed by the small business. The grant itself is converted from an STTR to an SBIR, and they provided $2 million to do additional R&D. Some other unique features of this funding opportunity, we are really looking at applicants who are less than 8 years from their terminal degree. We encourage women and scientists from underrepresented groups. There's a lot of emphasis on the mentoring component, and we expect that each PI will have both a technical and business mentor that they identify themselves. The application should also have very clear go, no-go criteria for fast-track transition. And with that, thank you very much for listening. I strongly recommend that if you're interested in any of these funding opportunities or in general SBIR funding, please reach out to the program office to get application guidance from us. Our contact information is present in this slide. You can also sign up for updates on our website, which is sbir.cancer.gov. Thank you very much.
Hello, and thank you for joining us. Today, I'm going to talk a little bit about an idea that we came up with a couple years ago to start to address some of the shortcomings of the biomedical research enterprise that have been limiting progress specifically for rare cancers and other diseases. The idea is a very simple one, and that is to go and create a new federal agency modeled on the Defense Advanced Research Projects Agency, or DARPA, that will address the biomedical research enterprise market failures. Now, specifically, this is being called the Advanced Research Projects Agency for Health by the Biden administration. We called it the Health Advanced Research Projects Agency, or HARPA. The name doesn't matter particularly. But what we want to get across to you is why we propose developing this agency. There are about 10,000 human diseases, depending on how you define a disease. And we only have interventions for maybe 500 of them. There's a massive market failure in the middle of the biomedical research enterprise. Current federal investment strategies by the NIH and other agencies have created the valley of death that has been limiting our progress and do not adequately address the market failures that live in that valley of death. ARPA-H would essentially do for health what DARPA does for defense. That is, to specifically create and deliver clinical solutions and new capabilities that raise the floor and allow private sector investment to drive the creation of solutions for different diseases, whether those be diagnostics or treatments or cures. ARPA-H would bring a different set of tools, approaches, and focus to the development of new platform technologies and capabilities to transform biomedical innovation. Through time-bound, milestone-driven innovation that has been proven effective through DARPA for years. And a little bit of history. DARPA was established in 1958. They are responsible for the internet, stealth technologies, radar, GPS, night vision, autonomous vehicles, and the Moderna vaccine. Indeed, it was DARPA, not the NIH, that made the initial investments in mRNA vaccines. They're designed to make pivotal investments in breakthrough technologies for national security and defense and directly address market failures that are impeding innovation for the purpose of national security. So the federal government can take the exact same approach and directly address the massive market failures at the center of the healthcare enterprise by establishing a new Health Advanced Research Projects Agency or Advanced Research Projects Agency for Health modeled on DARPA. The government has already created agencies with such capabilities for infectious disease with BARDA, energy with ARPA-E, Homeland Security with HSARPA, and the intelligence community with IARPA. So this is not a new idea. We're just applying this idea to healthcare specifically. Now, how does DARPA function? Well, it's based on a very simple set of principles that were developed by a former DARPA director named George Heilmeyer, and he crafted a set of questions known as the Heilmeyer Catechism to help agency officials think through and evaluate proposed research programs. And those questions are very straightforward. What are you trying to do? Without jargon. What is done today? And what are the limits of current practice? What is the new approach that you want to take, and why do you think it will be successful? Who cares? If you're successful, what difference will it make? What are the risks? How much will it cost? How long will it take? And what are the midterm and final exams to check for success? In other words, what are the milestones for success? Very simple and straightforward. So how would you take this particular set of ideas and apply that, and how would it be different from what the NIH already does? Well, they fund platform technologies and new capabilities. Okay, so they primarily operate through milestone-driven, time-bound contracts, not grants. They fund high-risk, high-reward research programs and development programs. The program managers make the funding decisions. If the project is not working out, funding is pulled and money is given to another effort. Program managers are not lifers at this agency. They would be there for 3 to 5 years, and there would be minimum bureaucracy, impeding them from making these investments. Now you compare that to the NIH. They fund basic science and clinical research through grants. They typically fund risk-averse and incremental projects, and funding decisions are made after peer review by academic research, and funding is rarely pulled. Career program managers make these decisions through highly bureaucratic mechanisms, and those mechanisms have led to the risk-averse approaches that the NIH has taken. So a little bit of our history. Now, we developed this idea about 4 or 5 years ago after the Suzanne Wright Pancreatic Cancer Foundation approached us and said, how could we actually start to address the shortcomings of the biomedical research enterprise as they apply to pancreatic cancer? During the Trump years, we had a tremendous amount of success, had support from the president, but the idea was ultimately killed because the NIH opposed it, because they saw it as competition with the agency for funding. We thought this was a shortcoming. So we wrote a white paper about it and put it out through the Day One Project and brought it to the Biden campaign, and they ran with it. They thought this was a perfect idea for addressing some of the issues that came up during the first cancer moonshot that was launched during the Obama administration. It went through the transition team, And it made it into the president's budget. The president talked about it in his first joint address to Congress, and most recently in the State of the Union address that happened just a couple months ago. Congress has now taken up the mantle, and they are going to fund the agency $1 billion. But they did something very, very unusual. They punted on the decision on where ARPA-H should live. Some people, some advocates like myself, believe that the agency should be independent from the NIH and live within the Department of Health and Human Services as an independent federal agency. Others believe that this agency should live within the NIH and be part of the NIH enterprise. That's where we are today. But how would ARPA-H complement the NIH rather than compete with it? NIH funds basic science and clinical research through grants. Grants are typically awarded to individual projects at academic institutions. We all know this. Collectively, these projects form the bedrock of our knowledge about biology, health, medicine, and disease. There is no question, without the NIH's funding and without the public's investment through the NIH, we would not be where we are today. However, that leaves this valley of death still. ARPA-H would provide the sustained drive needed to push through challenges and achieve medical breakthroughs by building these new platform technologies and capabilities based on the investments made by the NIH. And the Moderna vaccine is a good example of that. The investments by the NIH on the spike proteins for coronaviruses were pivotal in making the initial mRNA vaccines for COVID-19. But without the investment by DARPA in the mRNA vaccines, we would not be here either. It's the combination of the two that is really strongest. So this model means that diseases for which investments are risky or downstream profit potential are low are often ignored. There's often an underinvestment in platform technologies in particular. ARPA-H would step in where private companies do not, addressing market failures with direct investments that ensure that all patients have hope for a brighter future. And that sounds like a lot of rhetoric, but that's exactly what DARPA has done for the Department of Defense. So how would ARPA-H function? Contracts large enough to provide critical mass of funding to draw companies and academic research labs towards problems that are of a national interest. Minimum bureaucracy, a portfolio approach to high-risk, high-reward efforts where you're not just funding one project, but a number of projects aimed at the same problem. Government would control the contracts. If there's no progress, funding gets pulled and reapplied to another contract. No traditional peer review. The decisions are made by top-notch program managers, and there's a high turnover of those program managers. They're there for a short period of time and there to make a difference. The not-so-secret sauce is that all funding allocations would be left to the discretion of the ARPA-H director and the program managers. All funding oversight is entrusted to the program managers. And there is tremendous oversight on these projects. Program managers typically talk to labs a couple times a month. Funds would be awarded as milestone-driven contracts that give program managers the capacity for early termination, as we discussed. And this almost never happens with traditional grants for research and development. So where should ARPA-H live? This is the issue that Congress decided that they wanted the Secretary of Health and Human Services to make. There are strong arguments for it to be an independent agency and some arguments for it being at the NIH. At the NIH, it would be immediately connected to the other institutes and centers. And there's already a mechanism for managing this agency. And therefore, you might save a little bit of dollars. And you could spin it up a little faster. But that leaves the risk that this agency would succumb to the bureaucracy of a massive federal agency. Why take that risk? That's the question. So what happens next? The administration is going to make a decision on where this agency should live, and Congress is certainly weighing in with hearings and bills that to drive it to be an independent agency, and some to keep it underneath the NIH. This is where we're at today. But we are going to see an ARPA-H launched. That's the good news. Now we're in the details. That's a great place to be for the United States and for this administration and for biomedical research writ large. As I said, the president has been supporting this tremendously. He proposed a $6.5 billion budget to launch ARPA-H. Congress said $1 billion to start with. That's okay. So with that, I want to focus again on why we're doing this. We have this market failure in the middle of the system. The funds that we need to start to address those failures need to be spent in a different way than we've been spending it to date. And therefore, we need a new federal agency that will not succumb to the bureaucratic mechanisms of traditional biomedical research funding and really drive at change. And that change is critical, and the legacy it will leave will be extraordinary, the same way it has been for ARPA-E, for energy, and DARPA. So I sincerely thank you for your attention and support for this issue, and hope that I will get some great questions about how this could actually address rare cancers specifically. There's lots to do in this space. Thank you very much.
Those were great talks, guys. I listened to you guys and I have hope. And I'm going to ask the first question here. And can you guys all hear me? Just nod your head. Awesome. Since I'm the moderator and there's a number of questions in the Slido, and you guys can please, the audience can ask more. But one of the things that, and of course it's what was most recent. So Michael gave his ARPA-H talk, and I just want to, I hope those talks dovetailed well for the audience. They certainly dovetailed well for me. And it gets interesting now because I hope you guys can synergize and challenge each other and ad lib and be spontaneous. But I want to point out what Michael did. And every time I go to an AACR annual meeting, I'm just astounded by the complexity of the basic science that gets done, and it's just magical sometimes. But that what we're talking about, the translation of science, doesn't happen without the science first, and I think you pointed that out, Michael, as well. All the great work that the NCI does, all the great work that the NIH does. And I was at a talk just the other day. It was over the weekend. There was synovial cell sarcoma, which is only 900 patients a year. It was the SS18, SSX translocation. Its effects on the SWI/SNF, BAF, non-canonical BAF complexes, BRD9 degraders. If all that basic science wasn't done beforehand, and without that, I don't think anyone would have known to go after BRD9 with protein degraders. And so, as you pointed out, science comes first, and that science is in nematodes, yeast, and flies, and that insight is what can be translated into medicine for people. But my question, and as Michael points out, there's $1 billion out there for translation of science for ARPA-H, ARPA, HARPA. But what impressed me was Josh made the Chordoma Foundation happen, and no doubt he had lots of help. But he was a driver, and he was the passion for the Chordoma Foundation and for all the work that's been done. And Jeff Turetsky spent a career trying to drug the undruggable for EWS. Failed twice, kept going, and there are at least 2 kids alive that, in my experience, would absolutely not be here now. And Don has frequently brought up this point in discussions that I've had with him, is that there's a lot to be said to have ego in the game, boots on the ground, to be the team doing the work, to see the issues and hurdles, to go to bed at night thinking about them, and then wake up with a plan to tackle the day. And those in the lab sometimes have 24/7, 365 work lives. And DEPA funds founders of companies who desperately want their ideas to succeed, to sometimes the point where they've mortgaged their homes to try to support their companies. So Michael and the rest of you, allay my concerns that when I hear this idea of contracting out work efforts to folks, even when there are milestones and there's defunding, that those contractors, and granted, maybe DARPA is the point of, well, it did work, but the contractors may not have the same impetus and motivation that many of us have in trying to make science and medicine happen for patients versus a contractor versus an AACR scientist doing basic science. So help me out with that.
I'm not sure I understand your question.
I'm concerned that when we send for contractors, that it's not their life's mission. It's they do other things, and now they have a contract to do X, Y, Z. if it happens, it happens. If it doesn't, it doesn't. We lose some money, but we're going to go on. And whereas when your ego is tied up in it, when it's your baby, how much more motivated are you to do that and get it done?
Their motivation is to get something into the market, and so it's not just and also there is with scientists that work at companies, there is certainly the same kind of pride that you would see in someone who's doing research in a laboratory. So are you actually getting them to pivot towards something that they perhaps weren't considering before? Potentially, yes. But that doesn't mean it's any less important for them to be successful in that space. If they're going to try and bring something to the market, that is the goal of the company. And so it wouldn't be that they would just lose some money, but they would lose opportunity to bring themselves to market. And that's a huge motivator in the private sector.
Jeff, I would take it back a step. I would say that it's not enough. There are not enough brains working on these problems. And Josh may agree with me on this, that, the thing that I found as a PI in a lab for the last 25 years, finding people who are really motivated to really want to develop as independent scientists and become thinkers is a declining thing in this country. And for everything we're doing here to try to commercialize things, I think what's lacking, and this came out, I think, a little bit in Josh's talk, and others mentioned it as well, the creative ideas, the stuff to market. I mean, it's one thing to have an engineering process of something going through the pipeline, but we don't have anything in the pipeline in a lot of cases, in a lot of these tumors. And I think that, and this Mike sort of brought this out as well, that there is this sort of establishment view of how science gets funded. And Deepa brought up the fact that there's this new SBIR for not having preliminary data. But, every time I've been on a study section where no preliminary data was required, somebody always said, well, do they have any preliminary data for that? The creative idea. And, it's a great thing in practice. So I think there's some fundamental things. And I would say that at a certain level, people need to be brought into this process and encouraged. And I know this isn't exactly the purpose of this group, but I think that's something that we're really lacking.
Well put.
I would just
maybe I can go back to part of your original question. About skin in the game, boots on the ground. So Mike, the one thing I would say as a speculation, let's say, is that if ARPA-H can move very quickly and get these contracts out, it for certain kinds of projects, like an IND enabling project, IND directed project, it places a burden on the recipient, on the scientist, to essentially form an entire biotech company in order to get all the right people, all with skin in the game. So, which is a huge lift. And so what a lot of companies wind up doing is contracting out to a CRO or a CMO or using consultant. And I think in my mind, that's the skin in the game part that Jeff maybe is talking about, having a part-time consultant to do your tox, let's say, or to do your manufacturing may seem very obvious and natural, but I think our own work where we do have this boots on the ground team, I think suggests that you want a CMC partner or a tox partner on your team that's living, breathing 24/7, this project too. So almost think some kind of hybrid thing, where the ARPA-H grants get out fast, but there is also somebody for you to connect to that really will join with you, that already is an expert in some of the areas that you aren't, expert in to begin with. And that might be a nice hybrid system to go really fast, to go really hard, and to get the ownership embedded throughout the platform or process or whatever, all the way to the goal line.
Indeed. I don't know if any of you guys had the opportunity to listen to some of the big drug companies giving talks, over the weekend, but the DNA encoded ligands and the cassette chemistry that they do in a plate, and 100,000 compounds in one well, and they can screen for those and pull out a tool in a week and turn that into a reasonably sub-micromolar drug in a month, right? Those would be great platforms to have that ARPA-H would be able to provide for everyone to use, and that I'd be thrilled for that. That would be amazing, and I'm not sure how interested Merck and Takeda and BMS are going to be in sharing all their methodologies, but that would be fantastic.
Indeed.
Josh, of your experience of the last 16 years, no doubt you've probably run into obstacles and hurdles, but it seems like a major success to me that what you've accomplished, and I just wonder how you would guide somebody else. There are a lot of patient advocate groups out there, and they all work hard. But what would you say was the steps that made your Chordoma Foundation, and the fact that you have probes, if not real drugs, for brachyury?
Yeah, thanks, Jeff. Well, a lot of things. It's hard to boil it all down, but I think if I were to try, I think it's largely about being proactive and goal-directed. There's so many problems that need to be solved. There's so many inputs that are necessary but not sufficient in a research ecosystem to be able to make progress. And just putting money into a research ecosystem that's not optimized for speed, that doesn't have all the necessary components, doesn't go very far. But if instead you start with the end in mind and work backwards and think about what are all of the necessary components, and be proactive about ensuring that they're all in place, that the necessary reagents, that the necessary talent and investigators are all working in the ecosystem, you can go a lot farther than the kind of typical model of simply investing reactively. Certainly it helps to provide grants, but again, just providing grants without a clear plan, a clear goal in mind, only gets you so far. On the research front, I think another big lesson learned is on the fundraising front. Being proactive is a much more appealing proposition to donors. You might wonder, in a patient population of maybe 2,000 to 3,000 patients, are there enough people out there to support a meaningful investment in research? And I think the answer is, if you're just asking for money to fund unspecified research, probably no. But if there's a very, very clear plan in place, very clear research needs and priorities, what we've seen, pleasantly, surprisingly, is that there are people who come and see the value in investing there. And then I would say, with respect specifically to efforts around brachyury drug discovery, one of the biggest learnings came from a course that I took from Andrew Lowe at MIT on healthcare finance. It was really, really eye-opening. I think understanding the economics of what it takes to develop drugs is just completely illuminating, and serves as a guide to figure out what levers to pull. Where do what are the risks that go into the calculus around investing in a drug, and where are the opportunities? And if you can reduce the risk and increase the potential reward, we'll see if this pans out. But the conjecture is that there is interest and resources out there that will invest, if you can get that calculus right. And so that's really what's been steering us towards developing probes. If we can sufficiently de-risk this target, there are lots of companies, lots of investigators out there who have the capabilities to run with that, turn it into a degrader or molecular glue, for example.
So along those lines, brachyury, your I'm assuming there's some interest in this because it can generalize to some extent. And for the EWS Fly1, the Jeff, your drug, I'm a little confused, and maybe you can clarify to me how that apparently it works in other tumors, but they don't have the Fly1. It's not the helicase binding to EWS that's connected to the Fly1. So how does that your drug generalize? And just the whole idea of generalizing de-risking, but then and also tied in deep in here. But if you can't generalize and you have a really small patient population, 200, 300 patients, does de-risking really work? Does it, even if you show that the drug works and it's non-toxic, and let's say someone gets it through, one rodent species, is it really going to be attractive to investors?
So let me start by saying that some of the best science and the best discoveries were made off of rare models or very unusual situations that could be generalized. I'll give you a quick one before we jump into EWS Fly. There's a drug out there called JQ1. And JQ1 was developed using a cell line that we had prepared in our lab initially and shared with Chris French that allowed them to model the pocket for the binding of JQ1 as a blocker of BRD4 binding to acetylated histone. And that cell line came from a patient with a rare tumor that we called thymic carcinoma at the time. And that patient's tumor, it's very rare. And Jay took that concept in Boston and they changed the name to nut midline carcinoma. And then they started a registry, and then patients started coming out. So there were obviously more of these out there. But it turns out that was all extraordinarily rare. Like, this was just an incredibly rare thing. And here was a cell line that we got from this incredibly rare situation. And look where it took people. And all it took was using that underpinning of scientific knowledge to know where to go. So with EWS Fly, it turns out that, I feel like I've been a success and a terrible failure all at the same time, because the failure was that I wanted to make this drug really specific. I wanted to be able to make this drug so specific that it would do nothing else except turn off EWS Fly. And we spent time for the 10 years before that looking at antisense RNA to try to target the fusion region just so we could specifically inhibit EWS Fly. Well, it turns out that when you take a look at where this drug is potentially working, and we started to model it because, remember, we found it empirically by just seeing if drugs would stick to EWS Fly. That was something we were told could never happen when we started, was you'd never get a small molecule to stick to a surface of a protein. And of course, we proved that wrong. So that was the first thing. Could we even find a small molecule? And then it turns out a lot of our data shows that it seems to be affecting something in the Fly1 area. Okay, so along comes a paper by my colleague, Dr. Bertoni in Bellinzona, a number of years ago, saying that Fly1, which is, of course, the C-terminal part of the fusion, was important for lymphomas. So I wrote to Francesco and I said, hey, would you like to try our small molecule in your lymphoma lines and see if it works? And the answer was yes. Not only did it work, but it was actually, pretty potent. And now we're trying to open up trials with lymphoma leukemia. So it turns out that the initial attempt to look at the drug and where it goes, it looks like it's doing something with ETS proteins, which is why all those other papers in neuroblastoma and melanoma, they're all focusing on the ETS proteins, which are related. And so while it wasn't necessarily the plan to have it generalized, it turns out that nature gave us something that was relatively generalizable. Now, if you cross that with the fact that we've known for over 10 years that EWS Fly was intrinsically disordered, and that intrinsic disorder played a role in how the small molecule was interacting with these proteins, blocking the proteins. I think you called it a molecular disruptor, Jeff, in your talk. Okay, so now you've got this phenomenon of phase separation. Well, when I would mention disordered proteins and phase separation when I gave talks 10, 12 years ago, people in the room chuckled. They're like, oh, come on, what is this disordered protein thing? Well, now you've got companies that are capitalized with hundreds of millions of dollars looking for drugs that affect disordered proteins and phase separation. Again, it started off as this very rare scientific phenomena, and now it's generalizing. So I think you can never underestimate the power of making a good discovery in something, even if it's super rare, about what it might generalize to do.
Yeah, I just started to talk today, and it was on a transcription factor involved in one of the hemangioma tumors, and it was I think SOX18 or SSX18, but they hit on propranolol. Propranolol was being used in the patient, and they had a much milder phenotype than what they would have expected. And then it was a protein-protein interaction inhibitor that they just found serendipitously. I thought that was amazing. Deepa, does de-risking work?
So from the NCI's perspective, at least from the NCI SBIR's perspective, we realize that, it's a small patient population, and we're willing to fund we're very happy to fund companies, even though there is a small patient population, because we sort of realize that if we don't do it, the chances are less that somebody else would do it. So we're happy to take it a little bit, push it along further than we would other drugs. So we have that's sort of the perspective that we're taking. And as Josh said, if you sort of do good work in whatever one indication that you have, that works a lot more. We generally caution people about going and using the same drug in multiple indications, just because we rather see a lot more done with one indication, even if it is a small patient population, than somebody working in multiple indications.
So I want to make sure that we go through some of the audience questions here, and I'm going to read these. And one is for Dawn, for NCAT speaker. When reviewing the pitfalls and failures, please expand needs for training updated regulatory sciences for academics and early stage biotech staff, apparent goals in the EU by STARS Consortium, all indications, and then the US FDA Oncology OC by Project Catalyst. You guys do not want me to start talking about Project Catalyst, trust me. But I think this is more along the lines of, does the NCAT's involved at all in trying to mentor people? And that kind of what we have talked about before. We're trying to, have boots on the ground, ego, mentorship, guiding, versus just, talking to them twice a month and taking funds away.
Yeah, absolutely. I mean, we do our best to mentor. We have actually lots of ongoing meetings with groups like Josh's and many others, particularly at the very early stages, to try, just as Josh was just saying, try to give an idea of some of the other kind of superstructure or overarching concepts and motivators of the other industries. That would be critical for the eventual success of the drug, all the way through regulatory approvals and into the marketplace. And that could be on the business side, could be on the regulatory side, it could be on the scientific side in terms of non-academic science. So basically all what goes into an IND is hardly ever found in an academic setting. So we certainly try to do a lot of mentoring, but to do enough education and mentoring to make the academic PI or small biotech fully expert in all these areas in a time frame relevant to the clicking clock for patients, and on the business end, on the clicking clock for the IP, it's just not practical. So what we really tried to do is take this hybrid approach where all the experts you need to bring to the package, to the project to get to IND as fast and robustly as you can. We bring to the table. And indeed, the idea is not necessarily to do the lift all the way, but to de-risk the asset or the drug candidate enough so that you get into that next arena. So whether that's venture capital backing, foundation backing, partnership with a large pharma company. So in a kind of funny way, we love it the best as scientists when we help take a drug all the way into humans, and it does well in the clinical trial. But we try to refrain from doing that just because we're invested in the project and love the project. In fact, our greatest projects, in a way, are the ones that we've just decided to take on, and simply by the NIH deciding to commit loads of people, loads of years, millions and millions of dollars to the project. That in itself virtually de-risks the project to a certain extent. So a few of our projects, actually, once or twice every year, our projects, essentially, as soon as we commit to the project, that galvanizes the venture capital investment, and the project graduates early, so to speak. And while we had a little bit of heartbreak to lose the project that we had already come to be invested in, that's our ultimately, greatest, most efficient use of our funds and time is to de-risk as soon as we can, so that other sectors in the ecosystem take the project the rest of the way.
Jeff, can I make a quick
NCATS
comment?
have helped you? The two failures that you had, would NCATS have been able to help those failures?
That's an interesting question. I don't know. So, because I don't know fully what it would have looked like, but what I can tell you is that with the two failures, we didn't have the right team. When Scott came along and decided to invest in my ideas, he knew how to build the right team. And in fact, he did it mostly through, initially we were contracting out to various people, but he knew the right experts to go to, so that we went to a chemist who knew how to take and look at derivatives of the drug. We included a regulatory person, so that as the development was happening, the regulatory IND piece was being prepared. We had a toxicology person who knew how to get the right studies done. And it was absolutely critical to have the right people there, on the team. They didn't all have to be hired by the company. They weren't all full-timers with the company. In fact, most of them weren't. But they were all super experts, and they wanted to help. They wanted to be a part of what this project was going to be. And it would have never happened without that collaboration. And, I don't know, I can't really speak to what motivates individuals or what doesn't motivate individuals, but this was all about, people hearing the idea that I put out there, getting Scott's backing. And by the way, he was a business guy who really understands science. We had dinner with him very early on, and my wife was there with us, and she's like, oh my God, I've never heard anybody talk about your science with such knowledge. And this is a guy who's never gotten a science degree. He was basically a surfer at Santa Cruz. So anyway, I think it's all about how you build the team and who you put on it, and it doesn't matter where they are at the time. Hang on.
So Jeff, I totally agree. When that model works, it works fantastically well, and it gets the kind of commitment, exactly that you're talking about. If you take a step back a bit though, and you look at other funding programs from different places in the ecosystem where that's the model, that basically if you successfully get a slot in program X, then you get the funds to use for a CMO contract, for a CRO contract, and you get part-time consultants, assigned. And if you look at that and you take not a project by project, not a success story thing, but you try to do some metrics and say, okay, over 5 years, over 10 years, for the amount of money that you put into a program that's structured that way, this sounds like bragging, but basically our program is dollar for dollar, easily 10 times more successful in terms of getting projects across the finish line. So I think what it starts to remove is the risk, Jeff, that you got Scott, which is a magical catalyst, and it all worked. But not everybody can get their Scott, even with the same amount of money. And so in ARPA H, let's say, if there are ways to, I think the PM approach that Mike was talking about, I'll guess that part of the major consideration would be when you come to the table, do you have that team in place? And if you don't, do you have a path to get there really fast? In the venture capital world, putting the team together usually is milestone one, from angel to Series A.
The VC people that I've talked to say the team is so important. The science is important, but it's like they look at the team as well.
One other thing I'll add, and Don, this is you brought this up, and there's a little bit of a secret sauce in here that I didn't point to except in my acknowledgement slide. When President Obama created the Recovery Act programs that he did, and he gave a big tranche of money to the NCI, they gave it out in 4 groups: the RC1, RC2, RC3, and RC4. And RC4 grants were the biggies that came at the end with like all the money that was left over. And you had to have $500,000 a year of a budget, and you had to have something close to clinical trial. And so NCI gave out 2 of those RC4 grants. We got one of them. And we used the money. It was a $4 million grant over 2 years. We used that to do CRMO work with Battelle in Columbus, and they did the rat study I showed you. That rat study was $500,000. Okay, that one study took, because all the pilot studies getting to it, that was a $500,000 study. So that was really critical. So I agree with what you're saying, that if you can get things and get good value out of it, and I think that's what we did. So I had a chance to go to the White House at the end of, I was at 2000, right before President Obama's last year. I'm trying to think of that year. Anyway, and I got to actually thank him for making that act possible, because that gave us the seed money that really led us through the rat study, which then allowed everything to go forward.
Mike, there's a question here for you. How will the project managers be trained to bring insights about updated quality and translational regulatory sciences needed for tech and regulatory success? Were PMs also decision makers behind the costly failures by DARPA, et cetera? Everything's not perfect, right? We all make mistakes. I tell people that at FDA, we make mistakes and we try to correct our mistakes, right? And no one. So how no doubt DARPA has made mistakes.
Oh yeah, it's not a perfect agency, of course, and it's by its definition, it's high risk, high reward research. High risk research fails, but when you're going to fail fast and cut the funding and pivot to something else. This is it's taking the risk that the private sector can't take and that the NIH won't take. That's really what we're talking about here. In terms of the translation and the regulatory parts of this, they would some of the PMs would be brought in that already have those sorts of skills. But yes, they have at their disposal, they have the ability to hire consultants, and they have the ability to interact with other federal agencies. They have a few holds barred on what they're able to do in order to get up to speed with these sorts of things. So there's no one way to skin the cat. It depends on the situation is the short but probably unsatisfying answer to that question.
So as a program manager, it's not an individual, right? I mean, a program manager, however great they are, has a certain skill set, and developing a drug is a collaborative effort. So to Don's point, and the program manager has to leverage their network to get the right people to help the company. So that turnover of program manager means you're getting fresh talent, fresh network, fresh look all the time, and so that's one of the advantages.
Indeed. Well put.
One plug for Project Catalyst, Mike, is involve us with ARPA-H. We would love to be involved.
I'm not involved in setting up ARPA-H. I just proposed it and
the people. You know people I don't know.
Sure.
One other question there here, and we have like a couple minutes left, and we had an extra half an hour, which I am grateful that John Ratzloff got us. But someone was talking about various partnerships, financing, business, and they threw out the example of Bridge Bio, which I had never heard of Bridge Bio. But Deepa, maybe you know what Bridge Bio is?
I don't know which Bridge Bio this is. It's such a common term to have bridge and bio together. So I'm kind of confused.
So the whole question is, for all speakers, please comment on emerging options for new innovative business finance partnerships and risk sharing dedicated to commercial success and clinical access for rare genomic defined indications. One such example, Bridge Bio.
I'm not sure which ones they're referring to here.
What I didn't know was that SBIR is like the major funder for this kind of academic spin-off. If you're not going to SBIR, I don't know, as Deepa pointed out, there's, not a lot of other places to go with a lot of money.
Incidentally, I was just doing research on this a little bit, and DOD has just put out, I think, a funding opportunity for rare cancers research program under their congressionally directed medical research program, CDMRP. So that's I think it came out just last month or so. And so they have funding like $100K for concept all the way up to $2 million for more advanced projects. So that's a new funding opportunity that's just out there from a federal side.
All right, I want to thank
Jeff. I can maybe jump in on that question. I think the new innovative business finance models that are coming out, that are incubators or kind of semi-angel but more systematized angel types, or situations where some expertise comes along with the investment. I think those are clearly making a lot of sense for commercially viable drug products, because there's a lot of IP to go around, and if something gets big, then everyone can take it. over the long run and still be happy. And the scientist or the driver still gets a drug into clinic. I think what a bunch of us have talked about today is that for ultra rare diseases, cancers and non-cancers, that commercial framework is not in place. So there is not enough upside to drive that kind of dilution of intellectual property ownership back to the origins of the early stage. So I think they're good, but they're not necessarily relevant to the vast majority of diseases that have prevalence in the low numbers of thousands or hundreds or even a few.
Yeah, I am thrilled that you guys got to talk. I am excited that I got to hear Michael. I hope that NCATS somehow becomes part of ARPA-H, because I love the process that Don has. I love the team approach. But all of you are great. I thank you so much, and I thank the audience members who I can't see, but hopefully they are still there. And you're all going to go out in New Orleans and have those beverages. And I hope you guys have a great night. And thank you so much for attending.
Thank you.
Bye.
Thank you.
Thanks all.
Thank you, Jeff.